8 tonight, who knows? But anyways, for those of you who are here, let's do a warm-up. So in your notes, I want you to try and do or answer this warm-up activity on the electron transport chain. No bonus points, sorry. Usually in class, I kind of make it a race, but basically in your notes, I want you to have A, B, C, there's no D, A, B, C, E, F, G, H, I, J, K, L, and G here again, right? And I want you to try your best. I'm going to give you a few minutes to fill them in and label it. Label our electron transport chain. So I'll give you a few minutes to do that. Say the letter again. D. Just D is missing. There's no D. So there's an A, B, C, right? But no D. I don't know why. I just borrowed this image and everything's there. It's just, I don't know. They forgot G, or I mean D, I guess. How's everyone doing today anyway? All 13 of you. Are we hanging in there? Oh, goody. Yay. Yeah, same, I feel that. Ugh. I'm so upset that I'm online again next semester. It's such a drag. Where is everyone? I don't know. It's a great question. Everyone was gone yesterday too at my other school. Somebody thought it was maybe the World Series, or I scared them off with the electron transport chain. Who knows? That could be it, yeah. Well, that's why we're gonna take a look at it right now. So I'll give you another minute here. And then we'll go over it. Ah, that's a great theory. Well, hypothesis, right? It's a colloquial theory, not a scientific theory, but that's a great hypothesis. Yeah, journal's due tomorrow, so that could certainly be it. Oh, We got another person here, cool. Oh, 2 more people. Hey, oh. Now we're at 15, yeah. No way, we're at 17 now? I love that. Okay, ABC, there's no D, E, F, G, H, I. J, K, L. All right, well, let's go over it. So I'm gonna just kind of write out the answers here as we go along. So I'll actually keep this up. All right, well, here's our electron transport chain, as you can see, right, that we did last time and went through it in detail. So what was A? What is this? A is kind of referring to the space here, or the location rather. And yep, exactly. The intermembrane space, that was the space between the outer and inner membranes of the mitochondria. That's our intermembrane space. Awesome. Cool. B is our inner membrane, right? It's our mitochondrial inner membrane. That's where this is going to take place, this electron transport chain process. C, what was C? The area here. Yeah, that's the mitochondrial matrix. Center of the mitochondria. Awesome. Well, this slide should be available. Actually, it's just this picture that I have on a separate slide, actually. But I can send it to you guys. I can do that, actually. That's a great call. So let me write that. I'll send it in the email after today's lecture. Send. Fill in. Yeah. Yeah, because I have it in a separate one because it's a long story, but essentially because sometimes I don't use it depending on time, and you know, we're ahead of the game here, so I wanted to use it anyway. Okay, so we don't have D, so A, B, C, D, E. What was E? Yeah, no worries, no worries. Yeah, I think I'll absolutely send it. It's a great idea. So E would not be membrane proteins. It's referring to the electron carrier that arrives here. That's going to become oxidized. There's actually, well, it's the first protein. So what would it be? Yeah, NADH. So Grace, actually, it's subtle. Someone's joining, cool. It's NADH, it's a subtle minor detail, but FADH2, the other electron carrier that is full of the electrons and hydrogens, it actually starts its the process at the second protein. It drops off its electrons at the second protein first. The very first one, only NADH drops off. It's just a small minor thing, but yeah, we'll just do NADH for simplicity here. So NADH. And so NADH becomes oxidized, A, B, C, D, E. What's F? Yes, NAD plus, and you could even say the 2 electrons in the hydrogen. Absolutely. Yeah, might as well. Let's do it. I hesitated only because the electrons are going to bind and attach to that first protein, right? But you know that's the next step. Awesome. So NADH is oxidized. The electrons that are stripped off of that NADH bind to this first membrane protein to begin the journey down the chain, as we discussed last lecture. What is being actively pumped up? G. What is G? Yeah, hydrogen ions. Totally. So H plus hydrogen ions, positively charged hydrogen ions, or biologists sometimes just call these protons. Because really, that's all it is. It's a single proton being, and that's a hydrogen atom that doesn't have any electrons. It's a proton only. Okay. H. Ooh, H. What was H? Not FADH. So we're actually not going to put FADH in this diagram, but it's just another carrier being oxidized at the second protein. There's actually just not an option for it. But H is going to be binding to the last protein in the chain before this one, this unique one. Hint, hint, it's why we're alive, breathing. Oxygen, yeah, totally. So H is O2 or oxygen. The oxygen molecule will bind to that last protein. Okay. And what does oxygen become at the end of this process? Not ATP. So O2 oxygen is going to accept electrons at the end of this, right? It's our final electron acceptor. Water, exactly. And once it does, it actually is going to pick up 2 hydrogens to become more stable. And whoa, H2O. Totally. So we actually say oxygen is reduced because we give it electrons at the end of this, right? And hydrogens. It's reduced to water in this process, H2O. So oxygen's our final electron acceptor. Once it accepts these electrons, it takes on 2 hydrogen ions in the matrix and becomes water, and we breathe out the water that's produced from this process. Obviously, you don't really see it, but on a cold day, you absolutely can. It'll condense on the window and you can write your name or draw a smiley face. Okay, what was J? J is referring to this enzyme here. It's super spotty. Spotty? Oh, the sound? ATP synthase. Great. So ATP synthase is that enzyme. Not for me. Huh. Interesting. Well, basically, J is ATP synthase. And this was our protein at the end of the chain here that when the hydrogen ions were accumulating up here, high concentration up here and then lower down here, they're going to naturally diffuse from high to low by facilitated diffusion through this membrane, causing ATP synthase to spin. Does this ring bells from Tuesday? And as it's spinning, what's going on? So what would be K? What would K be? So this process is called chemiosmosis for sure, but K specifically is ADP and a phosphate. Yeah. So adenosine diphosphate, which is a molecule that has two phosphates attached, right? And then we're going to stick on another phosphate so that it's triphosphate. So what would that make L? What would our last one be here? Yes, ATP. Perfect. Awesome. That's the story. That's the story. We're churning out ATPs. And then here's the board if you want to write the answers down. And yeah, I'll send this out with the recording after today's lecture. The end product is not, so the product refers to the molecule that's going to be produced at the end of all this, or molecules if it's more than one product. So ATP would be the end product. Water would be an end product. And we could also say NAD plus, FAD are end products because they get oxidized. Their electrons get stripped from them. Chemiosmosis refers, that's the name for that process of when the hydrogen ions flow through ATP synthase. They just call it chemiosmosis. Okay, so before I forget, yeah, your journals are due tomorrow, as someone mentioned, so it's probably why there's some absences, right? But no worries. So there's that reminder. Oh, chemiosmosis is J through L. Let's see. So yes, but I would also include G playing a role there in chemiosmosis. So chemiosmosis, if you can see my mouse cursor, I think you can. What I'm circling here continuously, when hydrogen ions flow from high to low here and spin ATP synthase to produce ATP molecules, that's chemiosmosis, that final process here. Yeah, so that's chemiosmosis. And then the rest of it is the electron transport chain. So sometimes we call it, we just kind of say, oh, the third stage or the third part of cellular respiration is the electron transport chain and chemiosmosis. Yeah. Any other questions? Can anyone else hear me? That's no good. Okay, so Glorianne, so in my Zoom training, the best recommendation I have that they gave me, if you can hear me, so that's the other issue, So let me type bottom left microphone arrow. And then it's, there's a button, right? Where is it? Okay. Okay. Glorianne, if you can hear me, check out what I wrote in the chat. If you hit switch to phone audio, it'll call your phone and then the audio will go through there, and I think it might work better if it's a computer issue. So hopefully that works. Okay, to continue. So you know what's I wanted to show you guys? Some kind of good news, I suppose. So this is from our syllabus, and I was looking at it. And interestingly enough, what's today? The 22nd, right? Is cell respiration part 2 here. But we finished cell respiration. I was like planning to do cell division. So we're kind of ahead of schedule here, which is always good, because usually I'm very much behind. So today I'm really planning to kind of just review a bit, do some review on respiration, just kind of like that warm up we just did. And then maybe towards the end, start at cell division, or maybe even halfway. Yeah, but it's always nice to be ahead. You know, falling behind is no fun. And then of course the journal's due tomorrow. I did want to mention that. What else did I want to talk about? Let me see. Okay, let's do this. So we're gonna do some fun reviewing of sorts. We're gonna do the overall picture. We're gonna put it all together in the cell. So that'll be fun. We're gonna put it all together, the entire process of aerobic cellular respiration. A little better. Oh, good. Yay. All right. Overall picture. Okay. Just want to kind of connect everything. Bust out the colors. Oh, yes. The colors will be busted out. You need not worry. All right. This is, I really wish I had a giant whiteboard, but we're going to make it work. Draw a big cell. That was pretty good. That's our cell. All right. Here's a cell. And the first thing cells do after we eat, well, we break down our food, break down complex carbs, long polysaccharides. We make them into simple sugars and eventually glucose, which was our first starting material for glycolysis, right? At the very beginning. So glucose actually circulates in the blood and it's delivered to the cells in our tissues, all of our tissues. All of our cells need energy. So we have blood vessels everywhere, right? Delivering glucose from the food we eat. So let's draw glucose out here. It's kind of this ring structure as we've talked about. I'm going to put a G for glucose. And we mentioned when we talked about transport, this unit, how sugars, amino acids, these kinds of molecules are too big to just simply diffuse through that lipid, phospholipid bilayer membrane, right? So we need a protein. to do facilitated diffusion and transport the glucose into our cells. So all of our cells have these transporter proteins in their membranes, glucose transporter proteins. So we transport glucose in. Okay. So this I'll label as glucose. It's a C6H12O6. This is our glucose molecule. And the very first thing that's going to happen is this glucose is going to undergo glycolysis. And we're in the cytoplasm, right? So this is on the cytoplasm, right? The fluid inside of our cells. And that's where glycolysis takes place. So we're going to do our very first step, glycolysis. How many ATPs do we get from glycolysis? Perfect, 2. So we get the 2 ATPs here. I'm not going to do all the details. Actually, I guess I'll do a lot of them because that's what I have in my notes. Okay, that's better. So we get 2 ATPs. And we also oxidize glucose throughout glycolysis. It was like a, step 8, step 7, whatever it is. We're going to strip off electrons and hydrogen 2 times to generate 2 NADHs. So I'm going to start with 2 NAD pluses becoming 2 NADH. And for space purposes, I didn't include the electrons and stuff, but we're just kind of bringing it all together here. What was our end product of glycolysis besides getting ATP and NADH? What did the glucose ultimately become by the end of this? Two pyruvate molecules, absolutely. 2 pyruvates. All right. And now we're going to enter our mitochondrion, which I'm actually going to make my cell a bit bigger here, just so I can fit everything. And let's draw my mitochondrion here. It's the bean-shaped organelle. It has the inner membrane like this. Okay, now these 2 pyruvate molecules, these 2 3-carbon molecules are going to be transported, again, through proteins, but I'm just going to draw them straight into, I can include the proteins, it doesn't matter though, into the matrix of the mitochondria. So this is our matrix, right? And here, the two pyruvates are going to be converted into two acetyl-CoA molecules and this is pyruvate oxidation. So let me do, just trying to see what, oh, 6. Okay, so I combine things. Okay, so let's do, okay. This is what I'm gonna do. So my two pyruvates become two acetyl-CoA, right? During pyruvate oxidation, sometimes called the intermediate step or transition step. I'm going to combine things just for time and space sake, but we know from that pyruvate oxidation, we get 2 CO2 molecules and we get 2 NADHs. I'm going to add those into the totals with the Krebs cycle, which our acetyl-CoAs are going to enter. So actually for step 2, I'll do below here. I'm going to abbreviate pyruvate, PYR, oxidation, OX, plus Krebs cycle. On the slides, I kind of called it, or I did 2A and 2B. Here, it's just gonna be 2, pyruvate oxidation and Krebs cycle. And then let's do the totals. After acetyl-CoA joins in and undergoes Krebs cycle reactions, it combines with oxaloacetate, right? That was the first step of the Krebs cycle. But together, pyruvate oxidation, Krebs cycle, we get 2 ATP molecules. Let's do it. I'll do it here. 2 ATP. And then 2 CO2 from pyruvate oxidation, 4 CO2 from Krebs cycle. So that would be 6 CO2 total. So for CO2, I'll use purple. So I'll write it here. Plus 6 CO2. You can probably tell by now why I prefer having the big whiteboard on campus. So we get 6 CO2s from both of these processes total. 8 NADHs. I'm doing that in blue. 8 NADH. And 2 FADH2s. That was from Krebs cycle alone. And remember, the Krebs cycle, because there's 2 acetyl-CoAs, each acetyl-CoA undergoes those Krebs cycle reactions one time, so you do the cycle twice for both acetyl-CoAs total. And so each cycle you got 3 NADHs for 6 total. Get a close-up. Yeah, let me do that. I can also take a picture when I'm done of this and send it in the email as well. I think that might be a good idea. Is that 8 in front of NADH? Yes, it is. Sorry. Plus 8 NADHs. So you get 2 NADHs from pyruvate oxidation and then 6 from the Krebs cycle. So you get 8 total. Okay, so that's it for that. Cool. And then, last but not least, we have the ETC, which is going to take place, as we just discussed, across this inner membrane. Oh, also, let me, what's written on the arrow pointing to the second circle? Is it 2-acetyl-CoA? Is that what you're referring to? I can just read all of this. It's plus 2 ATP, 2-acetyl-CoA, because the pyruvate becomes acetyl-CoA. The second stage here, it's pyruvate oxidation and Krebs cycle, plus 6 CO2. This is our matrix, right? The center, where this is all happening. 8 NADHs total from stage 2 here, and 2 FADH2s. Again, this is like pretty broad overview. So you can use this to refer to absolutely, but when you go back to your notes, you can see the specific details, right? How we get these numbers. Okay, so CO2, small gas molecule, and we learned about small molecules and how they diffuse. They simply diffuse straight out of the cell through the lipid membranes. And so that's why we exhale them, right? All right. So the electron transport chain, the main things I'm gonna do here, I haven't used red yet. Oxygen that we breathe also travels in the blood. So here's the O2 up here, you can see that. And it diffuses straight to that final protein in the chain right before ATP synthase to be used to drive this chain, drive the electron transport, and is converted at the end, right, to water, which I'll do in blue, which also we exhale outside of the cell. And the last thing I'll do is that at this chain, our full taxi cabs arrive, our 10 NADHs, right? 2 here, 10 here. These are shuttled in as well. And then our 2 FADH2s arrive, and they're oxidized, as we went over, to become, so I'm just gonna write some arrows here, NAD plus, well, 10 NAD plus. I'll rewrite that. 10 NAD pluses and 2 FADs. So it's getting really small. So I'll definitely take a picture of this and send it to all of you. Roughly how many ATPs do we get from the electron transport chain per molecule of glucose? Who remembers the number? So far we're at 4 ATP. So 38 total. 38 total, right? 34 from this step alone. So we can get 38 total from this entire process. Absolutely. Is there anything I'm forgetting? I feel like that was it. Yeah, that's it. One more thing I do like to draw, though, is that because NADH and FADH2 are oxidized here, 34 from what? 34 ATP molecules. From the, just one glucose molecule that we began with can get us that number. So per molecule of glucose. Okay, so our NADHs and our FADH2 molecules become oxidized, right? And so this third stage, electron transport chain and chemiosmosis, we sometimes refer to it as an oxidative phosphorylation, as we talked about last time. And because these molecules are oxidized back to NAD plus and FAD, they can actually, well, we'll just show it with that NAD plus here, come back and pick up more passengers at the beginning and be recycled. Yeah, recycled, exactly. FAD can be used again in the Krebs cycle. So it's very important to what I call regenerate this redox potential, where we can do these oxidation reduction reactions again, where we're oxidizing molecules throughout this process to generate and reduce NAD plus to NADH by oxidizing a molecule in glycolysis, et cetera, et cetera. So it's very important to regenerate these molecules so we can do this again and produce more energy. Yeah, that's it. Isn't it beautiful? Let me take a picture. I can send it to you guys, like a close-up. Okay, I got a pretty good couple pictures there. Yes, Anna, that's correct. The 10 NADHs drop off their electrons and hydrogens during that ETC process and become 10 NAD plus, and then we get 2 FADs as well. Yeah, no problem. Okay, leave that up for a moment. Let's see what else I have here. So you guys are probably sick of watching videos, but this is a kind of a cool animation that kind of shows everything. It's 4 minutes. It's not very long, and I usually show it in class. So we'll watch this, and then we can move on to cell division. Let me, oops. Okay, yeah, so this looks really cool, and I usually show this in class, but like I said, you're probably over videos at this point, but let's take a look. Oh.
As this mountain bike.
Oh, okay, hold on. Sharing is paused, resume. Hello. What the heck? Okay, that doesn't work. Let me do this, hold on. Okay, here we go. While I play this, I'm just gonna go use the restroom really quick, by the way, sorry.
Biker heads up the trail. The breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, Energy in fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Yeah. All right. Isn't that a pretty good video? Yeah, I like videos like that because they're like only 5 minutes long, you know? So it's like, and it kind of cool animation. Okay. What's next? Oh, any other questions? Before we move on to cell division. I'm gonna send a picture of this, so. Okay, well, do I have my PowerPoint open? I hope I do. Should. Oops, maybe not. Let me see. Okay. Okay. Oh, is our quiz on what we just reviewed? Yes. Yes, it sure is. The lecture quiz. Yep. You reminded me. Yeah. So that's also due tomorrow. And it's on cellular respiration. Yeah. So the 5-point quiz won't have any of these questions. Yeah. Good question. Okay. So this is our next topic in this unit. Let me erase this. Today, what I like to do to begin cell division and the cell cycle is talk about a lot of new words and terms and concepts, because I've maybe mentioned before that biology is so much like learning a new language, in the sense that there are so many new words that we biologists use to communicate about life processes. And today, and this whole topic on cell division, there's a lot of new words. We kind of learned a few like in cell respiration, some fancier ones, like oxidative phosphorylation and all these things. But in this unit, there's definitely a lot of new words that you wouldn't really use in day-to-day life unless you're a scientist, a biologist. So we're going to focus a lot on that today. So let me pull that up. Load up the notes here. Shut my window. Loud. Okay. All right. These slides are, yes, they're on Canvas. Okay. So what we're going to be doing in this unit is taking a look at the DNA inside of our cells, more specifically inside of the nucleus of our eukaryotic cells. And we're going to be looking at them in the form of chromosomes. Chromosomes and also chromatin. So we're going to define these things momentarily. This is our genetic material. It is the information in DNA form, our nucleic acid DNA form, that codes for everything in your body. All the traits that you have is in this DNA form as sort of a, I like to say it's kind of like an encyclopedia of you, with all the instructions on how to create you. It's like a manual of sorts. And so let's talk about a few new words to begin. So we're going to look at, so we did the microscope lab, and we're doing it this week, right? And what you'll notice is cells look like this under the microscope. Right? They're circular, and then there's this darkly shaded region within it. So you'll look at the cheek cells in the lab, and you'll see these dark circles within the circles. This is the nucleus of cells, of our cells. And so what we're going to do right now is zoom in on that nucleus. We're going to zoom in and see what's going on. Okay. what's going on in there is actually this intensely dark stained, shaded region, whatever, looks like this, kind of like a bowl of spaghetti when you go up really close and look at it. That's what it looks like. And when it's in this loose form, this loose, stringy form, it's not very tightly packaged, right? It's, you know, like this, like spaghetti. It's called chromatin. So this is chromatin. So I'm going to write chromatin is a loose package of DNA and also proteins associated with the DNA. They're called histones. You don't have to know that. But chromatin consists of this loose, kind of like a ball of yarn or spaghetti arrangement of your DNA. Okay, now when you take a look at the slide here, you'll notice that it says chromosome, and this is what a chromosome looks like. And there's a coiled DNA molecule. So what are chromosomes as opposed to chromatin? Get another color for fun. So when chromatin, now notice I emphasize that it's a loose packaging, right? When the chromatin starts to condense and become more tightly packaged, they become these individual chromosomes, which is like a condensed, tightly wound up version of the chromatin. And specifically, there's going to be more than one chromosome in our cells. So I'm going to write chromosomes form when the DNA condenses and becomes tightly packaged around proteins, the proteins involved or embedded within the DNA. It condenses and becomes tightly packaged around proteins. Okay. Now, this won't make sense to you yet, but I want it in your notes so that you can refer back to it and kind of put it in the back of your mind at this time. But I want you to remember that during interphase, so we're going to talk about the cell cycle and how a cell spends its life during this lecture series. And during interphase of the cell cycle, the genetic material is in chromatin form. It's a loosely packaged form. Then when we learn about mitosis and cells dividing into two from one, at the very beginning of that, during prophase of mitosis, that's when this condensation of the DNA occurs, when the chromatin becomes more tightly packed. So this occurs during prophase. So I'll write prophase of mitosis. Now, the majority of this lecture series is focused on cell division. And so we're going to be looking at chromosomes a lot because mitosis is division of the cell, going from 1 to 2. But I think students sometimes forget, including myself when I first learned this, that 90% of the time, the DNA is in this state. And so I like to address that here. Okay. Chromosome terminology. So more new words coming your way. So what you're looking at on that slide is what's called a karyotype. Karyotype. And karyotype refers to what it is a visual display of all the chromosomes in a cell. Chromosomes in a cell. Okay. All right. So let's take a look at that karyotype. Point out some things for a human being. So any one of your cells go to the nucleus and notice they're condensed, so they're in chromosome form, right? So they probably took this during mitosis. Well, they definitely did when the chromosomes condense like this. And you'll notice here, there's 23, just ignore the X and Y for now. There's 23 pairs here, right? So in a human, all humans, every one of us has 23 pairs of chromosomes. So we have 46 total chromosomes. Okay, so the thing I really want to hammer today that I want to talk about is what's the important information regarding these pairs? Why do they look similar? What's the point of putting them in pairs like this? Why is it 23 pairs? Well, the first thing you'll notice is they're about the same length, right, for each pair. And however, you know, these chromosomes, right? Well, these chromosomes are different than these chromosomes, right? These are shorter. I like to use the analogy I mentioned in encyclopedia. So you could kind of think of our chromosomes from 1 to the 23rd pair as A to Z in an encyclopedia. And you're going to find different information on how to maybe produce digestive enzymes here on the 8th pair on one of these chromosomes. Actually, both. We're going to be talking about the differences and similarities. And then maybe on 11 here, you'll find information on how to produce antibodies or defensive proteins that our immune cells would produce to combat bacteria, and et cetera. It'd be different, right? So I like to think of it as an encyclopedia of sorts with different information throughout our chromosomes. Okay. So first thing I'm going to write here is that each pair of chromosomes is called a homologous pair. They are homologous chromosomes, okay? So let's write that. Each pair of chromosomes is called a homologous pair. They are similar to one another, or they are similar to each other, but not identical. And I'm going to talk about that at length. I'll probably talk about it again at the beginning of next class, because it's a very subtle concept. So they are similar to each other, but not identical. So for all of these homologous pairs, one of them comes from the father and the other comes from the mother, okay? So I'm gonna write one comes from the mother and we call this the maternal homologue. And then, of course, the other one comes from the father, and that's the paternal homologue. From the father. And that's the paternal, running out of room here, paternal homologue. Okay. So we have 4 minutes left. So that's actually, I know exactly where to stop, but let's take a look back at our chromosomes here. All right. So what I just wrote and what we just talked about, each one of these pairs here is called a homologous pair. And there's 23 pairs. What this means is that we get, and now I mentioned each pair one of them comes from mom, one of them comes from dad. So 23 total come from mom, and then 23 total come from dad, right? If one of each comes from mom and one of each comes from dad. So what am I saying? This is chromosome 1. You get 2 chromosome 1s, 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, 2 chromosome 4s, etc. All the way up to the 23rd pair. You're going to get one from mom, one from dad. So you get a chromosome 1 from mom, chromosome 1 from dad, chromosome 2 from mom, chromosome 2 from dad, etc. Now, how are they similar? How are they different? Let's address that to finish today. Actually, what should I say? Actually, you know what? I'm just going to throw out one more term before we do that. I think that, okay, I know what to do. Okay, so let's do here one more new word. For an organism, like a human being, that has 2 sets of chromosomes like this, 2 sets of 23, right, for 46 total, we call that a diploid organism or a diploid cell. Diploid. So di, right, is the prefix for 2. Kind of like disaccharide is 2 sugars. And so what a diploid cell refers to, a cell with 2 sets of chromosomes. So they have 2 versions of each chromosome. 2 chromosome 1s, 2 chromosome 2s, and next time we're going to talk about how they're different. How they're similar, these homologous pairs, but not identical. And emphasize on the not identical because they're different, right? Your chromosome 1 from mom is different than your chromosome 1 from dad in a subtle way. Similar, right? But not identical. Okay, I think that's a perfect place to end. So any questions about anything? My roommate's being loud. Questions, are we still here? Or are we ready for the weekend? Lab questions?